Since the Save Our Seas Foundation was founded more than 20 years ago, there's been tremendous strides in progress for shark conservation all around the world. There's been huge victories in international legislation protecting their trade. There's been massive growth in protected areas being created sort of just for sharks. There's an ever-growing community of advocates and passionate people that are dedicated to really trying to make sure that there is this difference being made for sharks and rays, and I'm hugely optimistic for the next 20 years and beyond, where I really believe that this growing global community can bend the curve, as it were, as one of decline for these populations back into recovery.
Well, hello, everyone, and welcome to tonight's Save Our Seas Foundation Distinguished Speaker Series. My name is Lance. I am the Director of Environmental Sustainability here at the Museum of Discovery and Science in sunny South Florida. If you ever find yourself here, come on down and visit our lovely museum. Tonight, we are going to be, we're hosting a wonderful speaker. Meghan Holst will be joining us talking all about the amazing world of Sevengill sharks and her research that she's been doing. Before we get started officially, though, I want to thank the Save Our Seas Foundation and congratulate them on 20 years of doing their amazing work on funding work on research and education and conservation on sharks and rays, and ocean health abounds. For tonight, I just wanted to go over a few little things on how tonight's going to go. For those of you that are new, welcome. We'd love to have you. We have a lot of returning viewers as well, so welcome back to those of you that are returning. If you have any questions tonight as we're going through, please feel free to put them into the chat. I'll be monitoring the chat for everyone and can relay questions to our speaker that way as well. We're going to have a dedicated Q&A session at the end, but if you have a question that pops into your brain as we're going along, just please put it in the chat and we can go back and forth with some conversation. A little bit about our museum. If you ever find yourself, like I said, in Fort Lauderdale, please feel free to stop on by. Museum of Discovery and Science is located in Fort Lauderdale. Our mission is to connect people to inspiring science. Our 4 strategic pillars here at the museum focus on physical science education, early childhood education, health and wellness education, and tonight what we're highlighting are environmental sustainability. So how can we improve and become stewards for our environment and learn about amazing research that is happening. So like I said, tonight we are joined by Meghan Holst. Meghan is a 4th year PhD student at UC Davis in the graduate group of ecology. Meghan has completed a master's degree master's in science in behavior and physiology of octopuses, with an emphasis on how these correlate with welfare. Now she's pursuing a PhD in ecology, studying the broad-nosed Sevengill shark in San Francisco Bay. Meghan is equally passionate about conservation science as she is social justice and science communication work. In 2021, Meghan co-founded 501c3 nonprofit Minorities in Aquarium and Zoo Science, and now maintains an executive directorship pursuing the mission to advance aquarium and zoo science by diversifying the professionals and their perspectives within it. Additionally, Meghan co-hosts a science podcast. You guys will all check it out, Sharkpedia, where the primary author of Elasmobreak, Sharks and Rays research, are interviewed to communicate their science strategies to the general public. Meghan intends to continue conducting science, communicating science in fun and interactive ways, and actively working to make the science fields a more diverse and inclusive place. Meghan, welcome. We're so happy to have you.
Thank you so much for having me.
Yeah, well, it sounds like you're doing a lot of really amazing work from working with elasmobranchs to your past work with mollusks and octopuses, and
Yeah.
works in helping the field be more inclusive. So kudos to all the amazing work you're doing. I know tonight you're going to be talking a little bit about Sevengill sharks, but maybe you can tell us about your podcast a little bit too, so we can all tune into that. It'd be really good.
Oh, sure. Yeah, I can start off with that. I was actually just recording a couple episodes earlier today. Are you ready for me to share screen?
Yeah, go ahead.
Great. Let's do that. Okay. So yeah, as Lance said, my name's Megan. My podcast is called Sharkpedia, like the encyclopedia of sharks. And I definitely would recommend checking it out. We have a lot of fun talking to shark scientists around the world and asking them questions about how they accomplished their research. And initially, this podcast was a really selfish way for my co-host and I to talk to other shark researchers as early career researchers ourselves. We had a lot of questions and not necessarily a whole lot of resources. So we use this podcast as a platform to connect with other shark scientists. And it turns out other people sometimes like listening to it too. So if you would like to learn with us, with all these awesome shark scientists, you can definitely check that out. Yeah, today we are going to talk about the broad-nosed Sevengill shark. So here in this profile picture of me, I have a baby broad-nosed Sevengill shark. I also call them Dalmatian puppies affectionately because they're so spotty. And I like to start off with a joke with this presentation, because a lot of people like to say these are shark-infested waters, but saying that there are shark-infested waters is like saying there are human-infested houses. They live there. Okay, so I want to break the ice with there are no shark-infested waters. There's just shark-rich waters, and they matter a lot. If you're here listening to this talk, you probably already know that and believe that sharks are very important to our environment. But not everyone necessarily knows that, and not everyone necessarily knows how many sharks are in the water. This is a picture near me. I'm in California, and this is in La Jolla, California, with a bunch of leopard sharks. Every year they have this huge aggregation. And if you step foot in the ocean, I can pretty much guarantee you've been in very close proximity of a shark, and they know you're there even if you don't. And they're not a threat. You have nothing to worry about. We really are the ones that are harming them, not the other way around. So sharks aren't infesting waters, but they do live here, right here in San Francisco Bay, where I'm lucky enough to have that as my backyard. Again, these are little profile pictures of the pups that we get to see here. Sevengill sharks are really cool. They are a deep water shark. You can see them on the left-hand side here. Broad-nosed Sevengill sharks are an apex predator. The females get a little over 10 feet long. The males are a little smaller, around 8 to 9 feet long. And they're in the family of what's called cow sharks. And they're called cow sharks because they quite literally become rotund swimming cows. This is an adult in this photo, but the really mature, especially pregnant females, just look like swimming cows. It's amazing how large they are and how agile they are as well in the water. And when a study was done looking at trophic placement, or also known as looking at the food chain, like where everyone falls in that food chain, Sevengill sharks actually ranked higher on the food chain than white sharks. So I personally am always putting a little shout out to Sevengill sharks, saying that they're hashtag the best shark. If you're on Twitter or Instagram, feel free to throw that hashtag on there and let them know that you're listening to this presentation and that Sevengill sharks are in fact the best shark. There's a friendly debate that happens online with other sharks.
They also have the most gills. So
they also have the most gills. It's exactly right. Scientists are not the most creative when it comes to naming things. This is the broad-nosed Sevengill shark is the full common name. And they're named that because they have a broad, wide nose and 7 gill slits, which is thought to be one of the most prehistoric lines of sharks. And as sharks evolved, they lost gill slits. Now, typically, sharks have 5 gill slits on either side of their head. The other really cool thing about them being prehistoric, this is a world map here, and all the blue regions are regions that we can find populations of Sevengill sharks. Now, our population is up here in the upper left-hand corner. ranging from northern Canada to, we think, Baja, Mexico. This population is not passing the equator and mixing with the South American population, which is not mixing with the South African population, which is not mixing with the New Zealand population, etc. So one of the questions I get is, okay, well, if they're not mixing, how did they get there? Well, like I said, this is one of the most prehistoric species of sharks. Sharks have existed on this planet longer than trees have existed on this planet, which is just a fact that is very hard for my mind to wrap around. But if you think about back in our like elementary school geography courses, our continents didn't always used to look like this. They used to be one big continent that we call Pangaea. And if you put those continents all back together, you can find Sevengills around what used to be the perimeter of Pangaea in temperate waters. And as those continents broke apart and shifted apart, those shark populations maintained in those temperate waters. So that's how they got there. They're a really prehistoric old shark. They typically live in really deep waters, around 200 to 300 meters deep is how long they're typically traveling between locations. And we get really, really cool access to them here in the United States. Not a lot of people hear about them, but here we have a profile of the west coast of the United States here. And what we do know about Sevengills is that we see really big, large, reproductively mature adults in Willapa Bay, Washington, which is a really remote bay up in Washington state. Humboldt Bay in the past has been mentioned before in literature as a potential pupping ground, and so has San Francisco Bay. So there's been some theories about, okay, maybe California is where all of these juveniles are living, and maybe up in Willapa where these reproductive adults are, maybe that's where they're breeding. There's a lot of assumptions happening, but not a lot of actual hard data supporting the theories that we currently have about this population. So there is a targeted fishery for Sevengill sharks here in San Francisco Bay. Every state has their own regulations, but I like to take a moment to say that, there's many communities that rely on fish protein as their primary protein source, and many communities, including especially island nations and including even some of our communities within the Bay Area, rely on shark meat as one of their proteins. So they might come and catch one shark a year, and then they freeze that and parse it out throughout the rest of the year. So there is a fishery for them, but that is something that we want to look at and evaluate whether or not that fishery can be sustainable and is safe for that animal's population long term. We actually get aggregations of them in San Francisco Bay, which is super, super exciting in terms of science. But if you're targeting a species in a highly concentrated area, that could end up being problematic. So there's reasons that we want to look into this fishery, especially these points of interest where we see them concentrated, to see what kind of impact humans are having on the population as a whole. The IUCN Red List of endangered species has designated Sevengill sharks as vulnerable, with the population decreasing. Now, that was a worldwide assessment made in 2020. But again, we really don't have enough data to look at even individual populations. There's not enough data with our population in particular to estimate population estimates, or really even defining exactly where the pupping nursery grounds are. That's been mentioned in the past, but hasn't necessarily been defined.
Meghan, can I ask a question about that?
Absolutely.
So I guess this designation as vulnerable, that doesn't necessarily afford them any special protections, because they're not listed on like the United States endangered species list or anything like that, even though they're vulnerable.
Yeah,
maybe reaching the more the endangered.
That's a really excellent question and point that you're making. The IUCN Red List is an international group that makes these assessments with experts around the world, but they don't have worldwide jurisdiction over other countries. So being on the IUCN Red List does not give anybody federal jurisdiction to do anything about that within their own country. So the United States manages our endangered species through the Endangered Species Act, and you have to provide really specific information to that group to demonstrate that an animal really does need resources allocated for the federal government to step in and say, yes, we're going to federally protect this animal and allocate resources to protecting it. That's when you start getting more funds. But to get an animal on the endangered species list is very difficult to do, even with critically endangered species on our coastline, like the taupe shark, also known as the soupfin shark. They're critically endangered on our coastline, and there's still an open fishery for them because we don't have enough data to demonstrate that they need to be listed on the Endangered Species Act. So that just takes a ton of funding, time, energy, and collaborations between groups to demonstrate that there's enough data for our government to actually step in for protection.
But like the work you're doing, the work that IUCN has done can help provide some of that data, I guess.
Yeah, exactly. It's a really great starting point. It gets people's attention, at least. IUCN Red List is internationally recognized. And so when the IUCN Red List puts in enough data and experts together in a room and says, okay, yes, this is a vulnerable species, it definitely matters. It just doesn't have legal jurisdiction over how we manage them.
Interesting. Thank you.
Yeah, you got it. Happy to answer those questions. I've had to figure out all of that myself as I've been doing this work. So Sevengills have been assessed, as I said, as vulnerable. So we think that their populations are decreasing, but that's been assessed on a worldwide scale. And if we want to manage this species, you really have to go much more fine scale and just look within populations. So we want to look at our own population of Sevengill sharks and manage that separately than how we would manage maybe another Sevengill population somewhere else in the world. So one of the questions that I'm asking that the Save Our Seas Foundation is helping fund to help answer is answering the question of, is San Francisco Bay a nursery habitat for Sevengill sharks? Now, it might seem obvious. I have a bunch of baby sharks here. There must be a pupping and nursery ground because there's baby sharks there. But there's actually really specific criteria that need to be fulfilled before you can designate something as a critical reproductive habitat, like a pupping or nursery ground. When I say pupping ground, that's just where the females come to give birth to the pups, the baby sharks. And a nursery ground would be where juvenile sharks stay for longer periods of time for either protection or higher food availability and resources. So oftentimes nursery habitats are areas where apex predators can't get in. San Francisco Bay, great area for nursery habitat because it is on average 15 feet deep for the entire bay, which a lot of people don't know. It's a massive bay, but it averages 15 feet. The adults are coming in under Golden Gate Bridge and swimming around Alcatraz and going back out. But the babies, where I'm finding the babies, are somewhere else, which I'll show you in just a moment. I'm getting ahead of myself.
So the adults are deep water and the pups are shallow.
Yeah, they're migrating at deep water, but the adults are visiting shallow habitat like Willapa Bay, Washington, and San Francisco Bay and Humboldt Bay. And in other areas too, like San Diego, we'll see them in kelp forests a bit shallower. So they come into shallow habitats, and I would assume that they're coming into shallow habitats for important parts of their life history, like breeding, pupping, or feeding. So if we're looking at San Francisco Bay, we have all these sharks concentrated here. And if we even want to talk about getting information to assess a population for consideration of the Endangered Species Act, for example, we need to actually define where critical reproductive habitat is, which includes where they're pupping and where their nursery ground is, which sometimes can be together, sometimes it's separate. Sometimes the pups can travel to their nursery ground. So my very first question is trying to fulfill this criteria to demonstrate San Francisco Bay is in fact a pupping and nursery ground. Now, this is defined by a paper by Heupel et al. In 2007. This paper was extremely foundational and is widely accepted across the entire shark community worldwide. So many publications have come after this, now using this criteria to demonstrate whether or not an area for a shark is a pupping and or nursery ground. So that's what we're using here for one of my chapters for my PhD. And that criteria is roughly first, where sharks are commonly encountered in areas other than other areas. That might sound really obvious, but it just means that you see a high concentration of them in this area. We see tons of these juvenile Sevengill sharks in San Francisco Bay, and you just do not find them anywhere else. You're not seeing them reported on social media. You're not seeing those reports with Fish and Wildlife. So that checks that fulfillment already. So we're trying to gather all the data to demonstrate. Here we're seeing this size class that we're not seeing somewhere else. And the second criteria being that sharks have a tendency to remain or return for extended periods. So maybe they show up and they give birth 1 year or for 2 years, but then maybe they never come back for whatever reason. Or, for example, another nursery ground for Sevengill sharks for the South American population, there's a bay between Uruguay and Argentina, and they have found that the juveniles are there every summer, but then they leave after the summer months. So they have actually defined this bay as a nursery ground, but only for certain times of the year. And then they don't know where they are for the rest of the year. Whereas in San Francisco Bay, what I'm hoping to demonstrate is that actually these sharks for this population are using San Francisco Bay all year round, and they're using it for extended periods of time. And the adults come back every year during that time when females are giving birth. The last criteria being that the area or habitat is repeatedly used across years where others are not. So again, that's not just showing up one time, but adults are coming every single year. Juveniles are consistently found here throughout the year and across years. So regardless of what time of year you're coming to San Francisco Bay, you can find juveniles here. And it's repeatedly used, not just for adults seasonally, but those juveniles year round. So that's the criteria. I'm gathering data at the moment to demonstrate that San Francisco Bay fulfills that criteria. So this is a rough map here of San Francisco Bay. San Francisco Peninsula is this white part right here that my mouse is over. Golden Gate Bridge is between these two land masses. This is Marin County up on the north end. So Golden Gate Bridge is here. Alcatraz Island is probably that little dot where my mouse is. And this is Angel Island up here. And one of the sites that I have been sampling, especially right now in the fall, is this site called Yellow Bluff. That is where the big reproductive adults are coming in. And this is the deepest part of the bay. So you'll remember I said this bay is typically 15 feet average around the entire bay. But right by Golden Gate Bridge, it's very easily about 200 feet. And where we anchor to go fish is usually between 90 and 100 feet. And so that's where we're getting these big reproductive adults. And previous studies have demonstrated that adults, when they're here, are coming, they follow the tide in and out of San Francisco Bay. So as the tide is incoming to San Francisco, they go under the Golden Gate Bridge into the bay. And when the tide goes out, they follow the tide back out, seemingly to follow prey in and out. And also, again, because females are coming in to give birth. And now the pups that we're finding are not anywhere by those big reproductive adults. They are way down in the South Bay. I know this scale doesn't look like much, but this is about 40 miles away. Right by San Francisco Airport is where we're finding these pups. And it is definitely a bit shallower here. The spot that I'm fishing them is actually the deepest part of the channel in South Bay, around 40 feet. But we think that they're scattered throughout this really shallow habitat. So this is where I'm sampling and not just demonstrating that, yes, juvenile Sevengills are here in San Francisco Bay, but where in San Francisco Bay? Because if we're going to even consider things like a protected area, you don't want to just put a huge blanket over this huge space where that might not be effective if you're covering a certain space where those animals maybe don't exist. For example, if we only put protections up here in San Pablo Bay up north, it's going to be a lot less effective if all the juveniles are down south, right? So I am trying to define specifically where these age classes exist so that we can demonstrate in the future, if we did need to consider protections, where to start, where do they actually exist, and what age classes do we need to protect. And the other question that Save Our Seas Foundation is funding to help me answer is how well Sevengills tolerate catch and release fishing. So like I said before, there is a fishery for sharks in San Francisco Bay. And a lot of times the fishermen will pull one up and they don't actually want that shark. They want a different one. So then they'll let that one go and try to get another animal to take home for food consumption. So one of my questions is, okay, well, when we're releasing them, are they dying anyway? Or are they actually surviving that pretty well? That's going to be really important to know, especially if this is a pupping ground. Are the females coming in and surviving that? Are they getting so stressed that they're giving they're giving birth to their pups in that moment? Things like that. Now, I will say really quickly, I'm not going over my third chapter today for my PhD, but my third chapter is actually working with these professional fishermen in the Bay Area because they actually are very protective of the shark population and they manage it themselves much more strictly than Fish and Wildlife actually puts on them. So in California, there's a restriction called a 1-bag limit for Sevengill sharks, which means that one person can take one shark per day. So if you have a charter of 20 people can take 20 sharks, and there's no restriction on the size, the sex, or the season of when you do that. The fishermen in the Bay Area know how long it takes for these sharks to get to this massive reproductive size, and they know that these females are coming in to pup. So they actually highly encourage and incentivize their clientele to let the big animals go back, and they take more moderate, non-reproductive individuals for their clientele instead. I think that's really interesting. I think there's a lot of misconceptions about who's participating in conservation and who's jeopardizing the conservation of a species. And I think fishermen are often categorized and demonized as individuals that don't care about the species, when in fact, I think they care sometimes the most about the species that we're talking about and have some of the most knowledge about them. So I said I wasn't going to talk about my third chapter, but I kind of did anyway.
That's okay. Yeah. Often the threats to these kind of, like, the real threats are commercial fishing and these kind of things instead of recreational fishing, and that can be true for a lot of different shark species, right?
That totally can. Yeah, and every species is different. Like I said, this species is usually 200 to 300 meters deep, so we don't actually have to worry about it quite as much in terms of being pulled up on trawls and things like that. But it is important for us to know if San Francisco Bay is a critical reproductive habitat where pregnant females are coming in and the juveniles rely on this habitat until they're big enough to join the big adults along our coastline. We need to know if this time when they're concentrated in our bay, if fishing for them and bringing them out of the water is also killing them. We have very special access to them, and so therefore they need special considerations for this area. So to do that, I take blood draws. This is a juvenile 1-year-old, young of the year Sevengill shark. So we pull up their tail and we'll take a blood draw underneath their tail. And we'll do that for a number of blood draws over a period of 30 minutes while that animal is actually swimming on the side of our boat, still on our line. So when we need to take a blood draw, we'll pull that animal up. We have water flushing over the gills so that they can ventilate and breathe the entire time. And we have specialized shark handlers that have been trained to help restrain them in a way that is safe for them and safe for us. We get a blood draw, and then we let them continue to swim on the side of the boat until that 30-minute period is done. And then we have a series of 4 blood draws over 30 minutes to basically see how their bodies are responding to that engagement. 30 minutes is pretty representative for an angling event. If you're fishing for a shark and it's fighting the pole and you're pulling it in. It's about 30 minutes roughly during those events. When you're fishing, you pull the animal out of water, maybe you take a picture with it, and then you put it back. So 30 minutes gives us a pretty foundational experience. Now, we are treating these animals as best as possible. So it's not perfectly a representation of what angling might be to them, because we're not holding them in the air. We have water flushing over their gills. But doing the most baseline in the safest way possible gives us a baseline foundation understanding of what their physiology is like under some of the best conditions when they're stressed, and then go from there. Some of these just initial parameters that we can read from the blood can't tell us the whole story about if they're surviving, but it is the first step to looking at how sensitive they might be to this kind of handling. And then it can be really revealing of if you need protections as well. That might also be different for a 1-year-old, like we're pulling blood from here, and a pregnant female. Maybe they have very different physiological responses. And so that's something I actually cannot look at for this project. I'm primarily looking at juvenile age classes, but I would love to look at in the future. And let's see.
What do you test for in their blood? Do you test for different hormones? I mean, I don't know if sharks have cortisol, but that's like the stress hormone in humans. But
Yes.
So do you check for different stress hormones in the blood?
Yes. This is a Pandora's box of physiology that I love very much, but very roughly, things that we're looking at are similar to some things that we would find in our blood during exercise, like lactate, glucose. Sharks don't have cortisol. They are primary circulating I can never say this word, glucocorticoid. Their hormone, their stress hormone, is not cortisol. Their primary circulating hormone is something called 1-alpha corticosterone. 1-alpha for short, because that is a mouthful. 1-alpha is very difficult to study. There's very limited access to studying it. I am working with a group to see if we can measure that for this study. But the world of shark physiology is, man, there's a lot there. There's a lot to learn. There's a lot we don't know. We're trying to compare it to systems that we understand, like mammals, and sharks just do not respond like mammals do. They don't have the same adrenal process. They respond to their environment very differently. They existed before trees, so their response to things is going to be very, very different than mammals, which evolved so much later in evolution.
It's really interesting. A common thing amongst a lot of the researchers that come on to the series is that there are still so many questions.
There's so many.
Yeah.
There's so much room in shark science. I'm going to put that out there.
That's where I was leading. Like, anyone out there listening, if you want to answer some of these questions.
There are so many. I feel like before I started in shark science, I would have thought, oh, all the shark science has been done. Like, there's no room for me. No. Shark science has barely been scratched on the surface, and maybe only for like a couple species. And there are over 500 species of sharks that we know of. So there's a lot of work to be had. But I want to mention, like, part of why this catch and release work is really important is because some species are extremely sensitive. For example, in Florida, if you pull up a great hammerhead shark, their likelihood of dying if their gills are exposed to air at all increases dramatically. So if you do pull up, if you realize that you have a hammerhead on your line, you have to cut your line. You can't even bring them up to the surface to take your hook out. Just cut the line, because their likelihood of surviving that interaction is higher than if you were to like pull them up, get them up on your boat, get them to the side, expose their gills to air, get the hook out. They actually might not survive that process. So before we can even know how to put any sort of management protections on how we're handling them, we have to know how they're dealing with being handled. We have to know how sensitive they are, because that's going to dictate how we can manage that species if they need to be protected in that way. So a lot of this work is very foundational. First steps because we just know so little. And like I said, my third chapter that I'm not diving into here is about trying to document the knowledge of the fishers along our coastline, because they're so incredibly knowledgeable. I am very excited for that chapter because there are very many things that are kind of like going to come out of that paper that are not documented about this species and that would otherwise go undocumented, that I think will help advance our understanding of this species and its dynamics. So if you're a graduate student out there, I highly recommend looking into social science as one of your chapters because it's very affordable. It's usually just your time and energy going to interview people, or like how to get there. If you have to fly somewhere, obviously that gets more expensive. But it's logistically much more feasible during a graduate degree than some like really larger projects like telemetry, where you're tagging them and doing like a 6-year study of their movement behavior. And you're supposed to be finishing a PhD in 6 years. So that can get really challenging in terms of timing. So I think social science is a great way to get a lot of information out there, and it's really accessible for people to do.
That's cool. Thanks for sharing. I just want to do a quick time check and reminder everyone. So if you do have questions, we are going to have a Q&A session in about 10 minutes. So I'll stop asking you questions and let you just go ahead and go, and then we'll save all the questions in.
No problem. We're actually really nearing the end of
cool.
This is the work that Save Our Seas has funded. And so like I said, this is very foundational work. There's questions of would areas of seasonal restriction support Sevengill conservation? I'm not there yet to answer that. We are so, we're in the rudimentary stages of getting really just like basic life history information about this species so that we can start to understand, do they need to be protected? How do they need to be protected? And where do we even start? So defining those really critical places of where they live is really important. So that if/when we're at the point of considering them for protections, that we have a starting point. And I say if/when because really species conservation is difficult. It's a difficult one, and I think our science community in general is shifting a lot more towards ecosystem-based management and trying to protect an entire ecosystem and not necessarily a single species, because really if one species in a certain habitat is in trouble, it probably means that a lot of other species are in trouble too. So there's a big shift towards ecosystem-based management that I think is effective and exciting. And studying apex predators like Sevengill sharks can help us identify some of those really important habitats where species are aggregated and are habitats of interest. So with that, I would like to thank my funders. I had Save Our Seas Foundation was my main funder for this work, as well as the Wild Animal Initiative and the Company of Biologists. Without them, this work absolutely would not be possible. You're welcome to follow me on any of my Instagram or Twitter accounts, and my email is there as well, meghanmholst@gmail.com. If you have any questions, you're welcome to reach out to me there. Thank you so much.
Thank you. It's really amazing work, and like you said, the species that has been around for such a long time and that we know so little about it's really fascinating. So thank you for your amazing work.
Yeah.
So we had a question come in a little early on about the age of the species of the shark. And just a reminder, everyone, if you do have questions for Meghan, please put them in the chat. This is now our Q&A time. The question was asking about the age of the species, and is it this? You had mentioned back, Pangea. Is it the same? Are they essentially the same species as they were 250 million years ago, or have they, slightly different? I don't know. Seemed kind of.
I mean, that's a great question. So I'll start by saying I'm not a geneticist, and there's maybe some technical things that a geneticist would want to say in answering this question. But I think the short answer is yes, it's the same species. I think every species has evolved, but I mean, Sevengill sharks, regardless of where you go in the world, they're very similar, genetically even. They're not closely related, but clearly had been connected at one point. And they're all the same around the whole world, regardless of the separation that they've had. So yeah, I would say yes, it's the same species. I just don't know how much they've evolved over time.
That's amazing to think about all these different populations of such a similar population of animals, totally separate. Usually when that happens, you have things change, right? Like Australia separated from those continents a long time ago, so you have all these unique animals there. But the Sevengills, even though they've been separated for so long, are still all the same. It's quite fascinating, I think.
Yeah.
Another question that came in just about the sharks. How long do they live?
Excellent question. I don't, short answer is we don't know how long Sevengills live. We're still trying to figure that out with a lot of species. Some species you can take, if you have a deceased animal, or if you sample a whole animal, you can take their vertebra and section that vertebra and actually count the rings of their vertebra like a tree rings to see how many years old they are. But not every species can do that, including the Sevengill shark. The way that their vertebra calcifies it doesn't form rings, so we can't age them that way. So
that's interesting, because like other some other deep water species like the Greenland shark, these kind of things, we know they can live hundreds of years. Would that be true for a Sevengill as well? Like, is it in the hundreds of years, or is it?
I wouldn't feel comfortable stretching it that far.
Yeah.
But I think it's definitely reasonable to think that they lived 80 to 100 years.
Yeah.
I don't know for sure, is the real answer.
That's interesting.
Maybe they do live very long.
Another question to be answered, right?
Another question to be answered. And another person, like, this is a bit outside of my specialty of how to age them when we can't use their vertebra. There's potentially some genetics that could be done to look at how old they are, but I'm just not familiar with those processes.
The little one that you had an image of that you were taking blood from, how old was that? That was a little shark, right?
That was little. That was only maybe 2 months old. That I do know, because we know how big they are when they are first born. And then we have there's been studies done demonstrating, like, when all the age classes are present, you can apply an algorithm that says, like, okay, if they're in this size bracket, they're this age. So that animal was born that year, that summer, for sure.
Right.
It's just when they get to their maximum size, then it's like, now how old are they? I don't know.
Old enough to reproduce. That's all that really matters.
Old enough to reproduce. Yeah. Yeah.
Oh, cool. So another question is asking about the ecological significance of apex predators like sharks in the marine ecosystem. How does their decline impact the overall health of the ocean?
Yeah, I mean, in general, losing apex predators in an ecosystem is disproportionately tragic. They have a disproportionate impact on the environment because sharks, apex predators in general, manage populations below them in the food web. When apex predators aren't there, other populations can kind of go out of whack. Like the next predator might come up and then decimate their prey species, for example. So apex predators are really important in terms of maintaining stability in an environment. So losing apex predators just invites stochasticity. It invites irregularity of our ecosystems, and it's harder to manage once you remove those environmental guard keepers. It's hard to catch back up once they're gone. So even if you did reintroduce them, you might not even fix the problem, because maybe the environment has shifted to that point where species interactions are going to be different. So conserving them in the first place is the goal.
Another question kind of related to that is, and there you had mentioned that the IUCN data shows their decline. What about in the California population? Are they better or worse? Are they kind of aligned with that? Or is there any way you have any inkling on populations
Yes.
are faring better or worse than the global average, I guess?
I don't know about the global average just because we don't have population estimates on, I think, any of the populations. But what I will say is that we have records in the US of decimating this population in the 40s. There used to be shark tournaments in San Francisco that targeted these big reproductive adults when they visited seasonally. And so for like, I think it was less than a decade, but in the 40s and maybe into the 50s, they had these shark derbies. And then fishermen reported not seeing big Sevengills anymore. So they shut down that fishery and they have slowly rebounded. So I would say like they've been, I think, a lot more stable in this last decade for sure. But in comparison to other populations, I couldn't say.
Yeah. Well, it's kind of on that note. What could we do to help? If anyone's interested, general public, how can we help this species? Or where a lot of our viewers are on the East Coast, but how can we learn more and help Sevengill sharks or other shark species? What would be your advice?
They can share this work. We need to talk about more sharks than the few species we see on Shark Week. Lots of them are, all of them are so important and underappreciated. I mean, this feels like a cheap answer, but it's true. Funding always brings access to this research because there's just not a lot of funding out there. It's getting smaller and smaller. Without Save Our Seas, this work wouldn't have been possible, truly.
Yeah. And I guess learn more, right? They can always they can follow you on your podcast and learn more about what you're doing and share that as well, right? What we mentioned, the connection to careers in the future. You started working with octopuses. How did you get involved with sharks and how did you end up studying what you were doing, and do you have any advice for anyone that maybe wants to follow in your footsteps?
Totally. So I'll say in general, like there's a bit of a joke between biologists that study sharks where we joke that there's like no such thing as a shark scientist. It's a made-up term because you can study anything and apply it to an organism, including sharks. So I was, investigating physiology questions in octopuses and then transferred that to sharks. And that being because I was really fortunate to have access to them at the time. And I had a ton of questions about why there were so many baby sharks in San Francisco Bay that no one had answers to. And so it just started, the scientific process of asking questions that we needed to investigate and actually answer ourselves. So that's how getting into shark science started. What was your...
The other question was just any advice to anyone that wants to follow in your footsteps in shark science or biology in general?
Yeah. Take any opportunity. Any opportunity is an opportunity to learn. And, my whole background has been in sculpin and like small fish and then octopuses. And now I'm applying that to sharks. So you can go to school for anything. My advisor is also a freshwater fish ecologist. He doesn't study marine ecosystems. He doesn't study sharks. And so I just want to be a voice of encouragement for people that are like, how do you get access? Access is difficult, but I encourage you to reach out to other people in the community. All of my research is highly collaborative with people across the United States and Canada. And they have made it really possible to give me access to things like measuring hormones that I wouldn't have had otherwise. So I would really encourage people to reach out and see, sometimes labs are collecting a ton of data, more data than they can use. So ask them if you can get involved and help facilitate using some of that data. Right. The other thing, maybe is less fun to hear, but there's a lot of jerks in this field. frankly, and towards minorities and towards early career researchers. And so one thing I just really want to make sure is said is that if you are early career and you reach out and someone's a jerk, believe them and move on. There are a lot of people in this field that aren't. So don't be discouraged. I've known a lot of people discouraged in this field if they've had negative interactions. And I just want everyone to know there is so much space for more science. There's so much science to be had, so don't give up. And anyone is always welcome to reach out to me on any of the platforms I provided if they want to ask more.
That's nice. Yeah, well, we could probably do a whole entire talk about your work with minorities in aquarium and zoo science, right? Is that a good platform for them to reach out?
Yeah, absolutely. That's my nonprofit organization. If you are interested in aquarium and zoo science at all, I strongly recommend reaching out and looking into our free memberships that provide professional development opportunities to our members, as well as some really great opportunities for just everyone in the industry, regardless of who you are. But they can reach out to me on my email that I had up there. I can put my slide back up if you'd like.
Yeah, please do.
Here's my contact info. Anybody is welcome to reach out to me on social media. I'm very slow to respond to. Email is the fastest way.
Awesome.
And here is the IG and Twitter accounts for the Sharkpedia podcast, as well as MAYAS, which is the acronym for Minorities in Aquarium and Zoo Science.
MAYAS, yeah. And I guess they can find Sharkpedia on any podcasting platform.
Correct.
Yeah. Okay.
Yes. We went on like a year hiatus, but we've been recording like fiends, and we're about to put out a bunch of new episodes.
Oh, nice. I can't wait. I'm gonna dig into it.
Yeah.
Sounds cool.
We have a good time.
Good. That's it for us. Thank you very, very much for joining us this evening and sharing your amazing work. And we'll have to have you back for the conclusion.
Oh yeah, I can't wait to get the conclusion. Hopefully very soon.
Right.
Stay tuned.
But yes, thank you again for joining us and for answering all our questions and sharing your awesomeness. We appreciate it.
Yeah, thank you so much for having me.
Yeah. On that note, next time, please join us for our last Distinguished Speaker Series event for this year, December 12th. We'll be talking to Janie Ray, CEO and lead researcher for BC Whales. So we're going to do a little shift in what we're doing next month for our finale, all about whales and whale research of British Columbia. So we hope to see you there, same time, 6 p.m. to 7 p.m. Eastern, December 12th. If you want to learn more, please visit us on our website. Thank you all very much for joining. Thanks again, Meghan, for being here and sharing your work. And thanks again to the Save Our Seas Foundation for making all this possible. Have a great night, guys. Bye-bye.